Hi everybody, so in this video I'm going to introduce you to lesson 8.1, where we'll learn about natural selection, and we're going to visualize natural selection using a JavaScript and then an HTML to better visualize it. So today we're going to combine the work of three scientists, Gregor Mendel, who is well known for his documentation of genetics and alleles. Specifically, we're going to be looking at allele frequency changing over time in response to some sort of selective pressure. Darwin is the one who is known for coining the term natural selection as it relates to descent with modification, meaning he understood that something was changing with each descending generation that modified the organism's or the population's ability to live and reproduce in its environment, which is what we call fitness. Darwin never got to read Mendel's work. They were doing their work at almost the exact same time. Mendel, did most of his work in the 1850s, and Darwin published on the origin of species and sort of his first papers on evolution or on natural selection or descent with modification around 1859. So these two were working at the same time, but they never actually got to communicate their work to each other. I also like to highlight sort of the lesser known Alfred Russel Wallace. He was a naturalist just like Darwin. He also proposed a natural selection theory to explain evolution, and that was sort of jointly published with Darwin's work, or published around the exact same time. But for whatever reason, Darwin is just more well known for his work. Darwin did a lot of his research around the world, but is specifically well known for his Galapagos Islands research. Alfred Russel Wallace, if you read his notes, his work centered around islands in Indonesia. But again, they both sort of observed the same type of behaviors, the same type of changes, the same type of features, and were able to propose this idea of natural selection. So let's focus on this term fitness as it pertains to natural selection. Darwin proposed these four postulates: that variation exists among individuals that make up a population, such as variation in size and shape, that some of the trait differences are heritable, meaning that they are passed on to offspring. Obviously, Mendel went much deeper to explain those patterns, but even Darwin knew that those traits were being inherited by offspring. Survival and reproductive success is highly variable. Many more offspring are produced than can possibly survive, so only some individuals in each generation survive long enough to produce offspring. And of those individuals, some produce more offspring than others. And then finally, the subset of individuals that survive best and produce the most offspring is not a random sample, but rather those that have the inheritable traits that are more likely to survive and reproduce. And here's a classic example of it. A population of mice arrives in a new area where the rocks are dark. We have natural genetic variation in the species, or in this population of that species. Some mice are lighter colored, some are darker colored. The lighter colored ones are more visible to predators. And so when we have this selective pressure of predation, those light colored ones are selected against, and the darker colored ones are going to be allowed to reproduce more, or be able to reproduce more, and pass on those traits to the next generation. So sort of a classic example of selection. And selection comes in three types. Here's sort of the original population with a gradient of color. We could select in one direction towards the darker color, which is what happened in that example that we just looked at, where the original population shifted towards the evolved population's direction. We could have diversifying or disruptive selection, where the median color, for whatever reason, is chosen against or selected against, and so the darker ones survive and the lighter ones survive. This could lead to speciation, or two separate species being identified. And we also have stabilizing selection, where the median is the one that is preferred, and the extreme phenotypes are selected against for whatever reason. So make sure you spend some time doing part 1, where we'll actually break down the types of selection and start to sort of see what the graph is looking at. In this case, we're looking at a different variable than color, which is body size, compared to the number of individuals in both directional, disruptive, and stabilizing. But what I really want to introduce you to is understanding the model that we're going to use, which is a JavaScript allele frequency modeling tool that lets us look at a single gene with 2 possible alleles. We'll call them P and Q, just like in the Hardy-Weinberg principle, rather than just a single polygenic trait like the body size. So this is a model that's more general and allows us to sort of model change in frequency of those alleles from one generation to the next, which is the basic example or basic definition of microevolution. So no mutation is occurring in this case. No gene flow is going on. There's no migration in or out of our population. We're going to assume for this model we have an infinite population size. And we're going to assume no genetic drift. Genetic drift is probably the hardest evolutionary tool or evolutionary pattern for students to understand. So I'm not modeling it yet for you. We'll model what this looks like next week. But we can assume for right now there are no, there's no random genetic drift occurring in the population. So in such a system, it's possible for a population to evolve in such a way that one of the alleles is lost. So let's say we had a big allele and a little allele. It's possible that if the little allele is selected against enough and the big allele is selected for enough, that little allele could be lost. And the larger allele could become fixed in the population. In other words, it becomes the only allele in this system. And so eventually the frequencies, if you think about Hardy-Weinberg, would become equal to 1, while the other falls to 0. And fitness is defined as the relative ability of an individual or population to survive, reproduce, and pass on their genes. So in order to understand this, we're going to define a couple new variables for our JavaScript. We're going to set a selection factor, S, where we are selecting against the homozygous recessive genotype. So that would also, in that case, select against the homozygous recessive phenotype. And then we're going to select sort of another variable, H, which is going to represent the selection against the heterozygote. In the case where heterozygosy shows an intermediate between the 2 alleles, that heterozygote individual could be differently fit than the homozygous dominant individual. So that's sort of one thing to think about, that if we have an incomplete dominance, if we had a co-dominant situation, etc., the heterozygote may be more or less fit than the homozygous dominant. In some cases, they would have equal fitness. And so our model could either keep this at 0, which gives that organism equal fitness to the homozygous dominant, or we could alter its fitness. And so here's sort of our fitness coefficients, the likelihood of a successful reproduction. Homozygous dominant individual, we're going to set at 1, just an arbitrary value. We picked 1 because it's the simplest whole number. The fitness of the heterozygote would be 1 minus the efficiency of the single allele it has for Q compared with its heterozygosy and the presence of P, or the dominant allele. And then finally, the fitness of the homozygous recessive would be 1 minus S, wherein we say that it could be more fit than the homozygous dominant or less fit than the homozygous dominant, depending on the value we set for S. So if S is between 1 and 0, what does that mean about the relative fitness of QQ genotype compared with PP? That would imply that its fitness is less than the homozygous dominant. So if we say 1 minus whatever that value is, 0.5, 0.3, whatever it is, we're saying we would set its fitness lower than 1. what would it mean if the value of s was negative? Well, if in this case, if we had 1 minus a negative s, you would add that s to 1, and you could increase the fitness of the homozygous recessive individual. So you may say, okay, this individual is more fit than this individual just by changing whether s is positive or negative. So let's use the model to actually show what we're doing. So first we want to download this file. It's called p_versus_time, where p is the presence of the homozygous dominant allele, or the dominant allele, versus time. So over time, many generations are passing, and we're seeing if the frequency of p in our population is going up or going down. If the frequency of p is going up, that allele is being selected for. If the frequency of p is going down, that allele is being selected against. So you should download it. I typically will save it to my desktop. So here I've got the file right here. And then you're going to click on it, and because it's an HTML file, it should automatically open in Chrome or Safari. So I'm going to click on it. And here are our number of generations on our x-axis, where we go from 0 to 1,000 generations. And then the frequency of p allele can occur anywhere between 0, where it's not present in the population, to 1, where it's 100% the only allele that is present in the population. And then we have many colored lines here that show sort of patterns of the frequency of p over time. So for example, this dark blue line here shows that the frequency of the p allele starts at 0.1 at generation 1, and over time eventually approaches, or gets to, if we ran out the generations, to be almost 100% present in the population, where it would be the only allele. And you notice that no matter what we set our starting p frequency to, all of these converge on this allele being the only allele present in the population. So some sort of evolution is taking place. So we use the axis labels to determine what the graph is showing. Why is the y-axis on a 0 to 1 scale? Again, it's going to be between 0% of that allele in the population or 100% that allele in the population. And the different colored lines are going to represent each possible iteration of selection for that allele. So based on that information, which of the 3 types of selection is occurring? Is it directional, disruptive, or stabilizing? Using the values of S and H given above. So we have set S to 0.1 and we have set H to 0.5, which means that our homozygous recessive individuals, if it's 1 minus 0.01 or 0.1, are less fit. And our heterozygote individuals have 0.5 multiplied by that 0.1, and so they're going to also be less fit. So the dominant genotype, PP, is the one that is most fit. And so no matter how many times we run out the iterations of this, no matter how many possible starting frequencies we see, P is being selected for every time. And so we see that it's shifting towards an extreme, and because of that, we can determine that this is a type of directional selection, wherein, let's say, P is the allele for large body size, we're shifting towards larger body size over time. So now we want to use a visual code editor where we actually see the code to open the same file and see and edit its code. So this is going to be a little tricky. You have to go to your file, right click on it and hit open with. You can open with text edit. I don't think that's great because it doesn't show you clear colored differences between the lines that you've written. So instead I'm going to go to Visual Studio Code. This is the program that I use. It's free. I recommend it. And that's going to show you sort of the whole code that we've written here that allows for this graph to be generated. This is a code that Christy and I wrote a number of years ago now when we took an online biological modeling course using computational modeling to show biological concepts. And there's a lot here. The bottom part, all of this HTML stuff, is just the information that the browser needs to actually chart the data. So that's visualization instructions for charting the data. You don't need to change any of that. None of that should be changed by you, or else this won't work. But we're going to be working with changing our variables a little bit. So this editable code that we're concerned with is the selection coefficient against the recessive allele, the selection coefficient against the heterozygote. Oh, I should change that to 0.5 because that's what it's supposed to be. Okay, I'll save that and run this again. Should be about the same, and it is, except selection happened a little bit faster, but it's still directional. And we can also set the number of generations. So I could show only 10 generations, I could show 1,000, whatever it is. In fact, I'll do that right now. I'll set 10 generations, save it, go back here, refresh. And now you see there's no agreement. If we go out to, let's say, 50 generations, save that, refresh. Now we start to see some agreement. If we go to 100 generations, save, refresh. Now we're starting to see P be selected for through direction. So I'll go back to 1,000 because that's what I started with. Okay. So if we change the value of S to -0.1 and reload our graph, we want to see if our prediction up here about the value of S being negative would come true. And remember, if we said S was negative, that would be adding a negative or subtracting a negative value here, which would essentially be adding a positive value to 1, which would make the recessive allele more fit. So I'm going to go do that. I'm going to change this to -0.1. I will save. Again, make sure you go File, Save, and then go back to your browser and hit refresh. Now we are selecting against P. Even if P is 99.9% frequent in the population to start with, over almost 200 generations, that allele will be selected against and become completely fixed out of the population. And the Q allele, which started, in that case, a really low frequency, is now going to be 100% available, or 100% of the alleles in that gene spot. So, what can we confirm? We can confirm that my prediction from part 2 is correct, and the type of selection that is happening is still the same as in part 2. It's still directional, but it's directional in the other direction. So now instead of going towards the large allele, we're shifting away from the large allele to the smaller allele, and thus we're fixing P out of the population and solidifying or fixing Q into the population. Okay. The initial settings were this. Using the values and most of the information, rank the fitness of each genotype in order from least to most fit. Okay, let's go back to our code, and we see that that's asking for the original, so I'm going to go back to this. And I'm going to say, okay, if S is 0.1, that means we are reducing the fitness of our allele by 10% every iteration or every generation. So our homozygous recessive genotype is going to be the least fit. If we add in the H coefficient or the heterozygous factor, we only look at sort of half of that genotype to lower the fitness of the allele, which means that heterozygous individuals would be sort of a median fitness, and obviously our homozygous dominant individuals would be the most fit for the environment. So I've ranked them from, sort of from lowest to highest in terms of fitness. So let's try increasing the value of H above 1, leaving S at 0.1. We can change the number of generations if we need to, so let's see if we need to do that. So we want to increase the value of H above 1. And I'm not going to go too far above 1. I'm going to go 1.01, something like that. And again, I will save and go back here, and I will refresh. So this is interesting, right? We have agreement that P is being fixed into the population as the only allele, but the time that it takes for that to occur is now much greater than it would have been otherwise. And the stronger we make this, the quicker it happens. And it doesn't necessarily always happen if we create a fitness that is really strong for the heterozygote. If we create a fitness that's really strong for the heterozygote, sometimes the Q allele is chosen and sometimes the P is chosen. And so we end up with this pattern where we're fixing one versus fixing the other. So the question here is describing sort of what happens to allele frequencies. Up here. So that is saying that we see that sometimes P is selected for and sometimes Q is selected for. Depends on the starting frequencies of those alleles, which would imply that some sort of directional or disruptive selection is occurring, where sometimes we're choosing for the dominant allele, sometimes we're choosing for the recessive allele. And because of that, we're sort of doing some sort of disruption. What does that mean? That must mean my heterozygote is now the least efficient, or the least fit. My homozygote recessive is median fitness, and my homozygous dominant is the most fit. So we have a disruptive selection occurring. So now let's make H negative. So this is going to be a little bit tricky. We're going to go to minus 0.5. Now I'm going to save. And now we're going to go back here and refresh. Ooh, what is happening? So no matter what the starting frequency of P, we are selecting for it, but it never is fixed. It never ends up being the only allele in the population. Some portion of the population is still showing the Q allele. So the frequency of P allele becomes fixed at 0.75 in this case. So 75% of our offspring, of our population, 75% of alleles in our population are P, or are dominant. So rank the fitness. Okay, let's rank the fitness. That means that we've given the fitness coefficient for H to be higher than the fitness coefficient for little q and little p. So our least fit is Q, or homozygous recessive. Our median fitness is homozygous dominant, and our top fitness is heterozygosy. So what kind of selection is occurring in this scenario? Well, we're converging in the middle. And so that one's pretty easy. We can go back here and say this must be a stabilizing selection, where the median body size, the heterozygote, is being selected for. Great. Make a prediction about what would happen if H was 0, and explain by connecting relative fitness of different genotypes to selection, direction, and type. Does the speed of evolution differ from the first simulations you ran? Explain why. Okay, so now we want to say that the heterozygous factor is 0. Okay, I'll save, refresh. And we still seem to have fixation towards P, where P becomes fixed no matter how many times we run this. The pattern remains the same. Did the speed of evolution differ from the first simulations you ran? So in this case, we've made the fitness of our heterozygotes equal to the fitness of our homozygous dominant. And the only fitness we've disrupted is QQ. because we multiply 0 times s, which essentially removes that, and so the fitness of p cubed remains the same. So it may take a little bit longer for the evolution to run, but essentially it's still happening, right? It's still occurring. In line 28 of the code, the equation that determines change in p, so the change in the frequency of p from one generation to the next, is written out. So let's go find that code. So here's line 28, and here is the equation. Delta p is equal to this divided by this, where in p and q and s and h are all represented in some way. So we can write this out, we can look at it, we can try to understand it. It's a fairly complicated formula. You don't need to know what's going on in there, but you do need to realize that this is based in Hardy-Weinberg. In other words, we look at our selectivity of our heterozygote versus the selectivity of our homozygous recessive versus the selectivity of our heterozygote. So, you don't understand the sort of equation, how this is arrived at. And to be honest, even though I wrote this code, I've forgotten myself exactly how they come up with this. But it's the code that's going to be determining how p changes from one generation to the next. So why is this equation so important? By putting it in your own words. What is delta p? So the change in p from one generation to the next, which means the original p, the p in one generation, is going to be p plus delta p. So if delta p is negative, p is less frequent. If delta p is positive, p becomes more frequent. And so each generation that goes by, each iteration of generations that runs by, we're going to change p a little bit, and then over thousands of generations, we can see what has happened here. Okay. So I'm going to stop there and give people time to do this last part, which is to sort of think about real-world examples and what type of selection would be associated with these particular examples. If you still have questions on this, feel free to come to office hours. It's a really simple but useful tool for understanding selection over time and easily being able to manipulate a model to demonstrate directional selection, disruptive selection, or stabilizing selection. Thanks.